我2016年嘅第8條題目啦，又嚟到科學探究咯。咁過往呢，其實都有問有關於科學探究嘅題目噶啦。咁啊，都係有關於做下實驗啦，俾個實驗嘅初衷你啦，俾啲實驗嘅結果你，等你去分析下啦，從而啦去得出一啲結論啦，或者啦去考下你成個實驗啦，點解要做呢一步啦，從而咧就引申一啲科學嘅本質咧去問下你嘅。今次整個實驗咧就有關於遺傳信息嘅。咁喺30年代咧，就有一位丹麥嘅生物學家 Hammerling， 往後咧我就即管就叫佢做錘仔先啦，因為 Hammer 就係個錘啊嘛。錘仔哥咧就想去探究一樣嘢啦，究竟一啲真核嘅細胞，佢會將一啲遺傳嘅信息儲存喺邊一度呢？咁佢就揾咗一啲單細胞嘅藻類咧去進行佢嘅實驗啦。咁下面啦就係佢所用嘅2款藻類啦。而呢個單細胞嘅藻類佢個形態咧，就同我哋平時見到一啲單細胞生物，例如阿米巴變形蟲啦，又或者啦，我一個嘅紅血球啊，一個白血球啊，一個葉肉細胞啊，係有啲分別嘅。佢分別有底部啦、柄啦同埋頂部嘅。而呢款單細胞嘅藻類咧，係有2款嘅物種，一款咧就圓形頭嘅，另一款咧就花花頭嘅。咁成條題目係會問啲乜嘢呢？嗱，呢度咧又教大家少少技巧先啦。有陣時咧做呢啲實驗題目咧，唔好咁快睇啲資料住，好似通識咁樣睇咗佢問你乜嘢先，跟住 part A 佢就問啦，要我哋描述下以上呢個實驗嘅結果。咁我哋呢度咧就有2個嘅實驗啦 ，Treatment 1同埋 Treatment 2啦。咁我哋入題目咯噃，成個 part A 咧。其實就係考緊我哋全視技能啦，我哋點樣去直接描述翻喺呢個實驗嘅結果呢？我哋觀察到啲乜嘢呢？睇下個實驗做乜先。首先啦，佢就將呢個物種1咧，呢個圓形頭呢個呢，就將佢斬開佢。佢有2款斬法。第一款呢，就係將個頭斬咗去，冇咗個頭咯。第二類呢，就斬咗個底部，冇咗個底部咯。從而就會觀察翻所剩餘嘅部分 remaining part。究竟有冇一個再生嘅現象出現啦？咁睇下結果咯。Treatment 1咧斬咗個頭係會生翻個頭嘅，而 Treatment 2咧斬咗個底係唔會生翻個底嘅。呢個就係我哋嘅結果啦。咁點樣寫呢？好多同學咧面對呢啲 description 咧，其實係覺得好困難嘅，因為喺書學唔到啊嘛，係要即刻望個結果即刻寫啊嘛，就寫到一塌糊塗啦，唔知搞乜好啦。所以呢度咧，梁 Sir 想提翻大家。答題目一個好簡單嘅概念，用短句作答。之前都拍過段片，有關於讀 Bio 嘅致勝關鍵同埋致命傷害。其中一個致勝關鍵就係用短句作答，用最簡單嘅語句去答呢一個問題。究竟我哋嘅實驗結果，我哋睇唔睇得到？cut 剩咗嘅部分有冇一個再生嘅情況？斬咗個頭，生得翻個頭；斬咗個底，生唔翻個底。你點樣形容呢一件事呢？成個框架就係考我哋點樣去全面嘅描述。乜嘢叫全面描述？有2部分。第一個部分就係成個實驗你做咗啲乜嘢？阿錘仔哥究竟你做咗啲乜嘢呢？第二部分就係個結果啦。咁究竟阿錘仔佢觀察到啲乜嘢呢？所以啦，第一句咧就好簡單嘅啫。喺 Treatment X 即係 Treatment 1或者 Treatment 2啦，Y 係邊一個部分係俾人哋斬咗去啊？而第二啦就係結果啦，阿錘仔觀察啲乜嘢呢？ It was found that 發現得到乜，又或者我哋能夠發現得到點呢個 X Y Z 照翻住幅圖嚟描述，你就搞掂噶啦。所以今次我想講嘅就係唔好跟佢個 model answer 啲英文咧好艱澀嘅，就算你用中文寫，你都會寫到一塌糊塗嘅。咩 Those with the cap removed could regenerate the cap。咩又斬咗個頭，又生翻個頭。喂，梁 Sir，我英文已經差噶啦，點寫啫？我教你用簡單英文，即使你將佢變做中文，都係咁樣寫。第一個實驗點樣形容啊？物種1嘅頂部係被移除咗嘅 ，the cap from species 1 was removed。然後啦，講結果咯 ，it was found that the regeneration of the cap occur。照抄啫嘛，去觀察一下有冇任何嘅再生去發生嘅。有啊，個頂咧係再生咗嘅，咪照抄題目寫翻咯，同佢 model answer 嘅呢一句一模一樣。你諗下隔籬呢句你點寫？咩？如果有個柄同埋有個底部嘅話咧，係可以再生翻個頂部出嚟嘅。你講乜嘢？我隻中文字都識，溝埋晒我就唔知你講乜。第二個 treatment 點寫？In treatment 2，第二個實驗咧 喺呢個物種一嘅單細胞組類呢，我斬咗佢個底部，而個底部係冇再生嘅。In treatment 2, the foot from species 1 was removed, and it was found that the regeneration of the foot did not occur。照抄題目，短句作答就是這樣。好多同學硬係覺得我明明識㗎梁 Sir， 但係我寫唔到啊。我俾嘅建議就係你要複雜簡單化，而唔好簡單複雜化。下面個 model answer 就係簡單複雜化。我評估俾你呢，就係 model answer， 我睬佢都有味。我寫呢兩句一模一樣意思，一定有分攞。好，去到 part B 啦，咁啊根據翻頭先 A part 嘅實驗啦，咁啊徐仔呢就有嗰個假說啦，佢就話啦，斬咗個頭啊，會生得翻個頭嘅，但係斬咗個底呢，就生唔翻個底出嚟嘅。有個假說啦，啲遺傳信息呢，就係儲存喺個單細胞組類嘅底部嗰處嘅。其實你睇題目呢，都知少少端倪噶啦。其實喺呢兩個嘅單細胞組類嘅底部有細胞核嘅，咁細胞核入面儲存住遺傳信息都好合理嘅。徐仔咧都冇諗咁多，嗰啲遺傳信息係咪喺個細胞核入面，我都唔理咧，總之喺個底部啦。咁跟住啦，佢就做多咗一系列嘅實驗，就想去研究一下遺傳信息嘅表達啦。咁佢做咗啲乜嘢呢？唔講住，又係睇咗題目先。我哋先解決第一 part。當阿徐仔做完呢啲實驗之後呢，佢有個簡單嘅結論啦，就話呢，移植嘅時候啦，被切斷嘅病仍然含有嚟自物種一底部嘅短暫指令，導致到第一次再生呢嘅頂部呢呈現一個混雜嘅形態。混雜形態咩意思呢？究竟佢做咗啲乜嘢呢？從而咧就睇翻呢個單細胞組類嘅細胞入面，究竟有一款乜嘢嘅生物分子啦，係呢一個短暫嘅指令啦。而呢一個短暫嘅指令係點樣影響到呢個頂部嘅形態呢？我哋睇翻實驗咯。阿徐仔你做咗啲乜嘢呢？第一步啦，佢就將物種一嘅病就同物種二嘅底部去移植結合埋一齊 ，grafting 呢個叫移植啦。移植咗之後望一望佢哋個頭生成咩樣咯？又唔似1號樣，又唔似2號樣，係一個混合形態。白你乜水嚟嘅呢？係咪一種新嘅嘢嚟噶？唔知啊，再斬咗你個頭，再做過。因為根據第一同第二個實驗呢，我哋知道啦，斬咗個頭呢係應該生得翻個頭出嚟嘅。咁我睇下你咩葫蘆賣咩藥咯。生完之後發現，係變翻2號仔，變翻呢個花花樣，究竟咩意思呢？做完成個實驗，咁我就睇翻徐仔呢一句呢，就知道有啲嘢啦。呢個短暫指令究竟係乜嘢呢？其實都係考緊我哋有關於一個短暫嘅遺傳信息，其實就係考緊我哋信使 RNA 嘅概念啦。去到 Part 2就問翻啦，信使嘅 RNA 究竟點樣影響一個頂部嘅形態呢？其實就係考翻 mRNA 嘅重要性啦。呢兩步呢，就不得不提噶啦 ，transcription 轉錄 ，translation 轉譯。今次係講緊mRNA 嘅重要性，所以我哋將個重點擺咗喺 translation 嗰處啦。究竟 mRNA 經過轉譯製造嘅蛋白質同個單細胞組類個頂部個形態有咩關係呢？信使 RNA 就係去指揮翻蛋白質嘅製成，透過咩步驟啊？轉譯咯。蛋白質有兩個角色，一個呢係功能性嘅角色，例如酶啦。另一個咧就係建構性嘅角色嚟啦，點樣形成一個花花嘅形狀或者一個圓形頭嘅形狀啦？呢個就係信使 RNA 蛋白質同埋形態嘅關係啦。到 Part 2咧，我哋就要去講翻究竟實驗結果係點樣去支持翻阿徐仔嘅假說呢？咁阿徐仔有咩假說啊？遺傳信息係儲存喺組類嘅底部。係真唔係啊？所以咧佢就做咗第3個實驗，將個病咧同個底部咧結合，咁睇下生出嚟咩樣咯。徐仔你話啊嘛，徐仔老老實實啦，你講噶嘛，遺傳信息係儲存喺個組類嘅底部。如果呢一句係正確嘅話，出嚟個頭咧應該係屬於2號仔，即係話一個花花樣。如果呢句係錯嘅話咧，信息咧就應該唔係坐喺底部咯，咁應該坐喺個病啦。個病係1號仔嗰度嚟噶嘛，所以啦，呢個混種嘅頭部應該係圓形頭嚟噶，咁啊死啦！佢又唔係圓形頭，又唔係花花頭，係混雜形態嚟噶。你幻想下，如果你係徐仔嘅上司，你督導住阿徐仔去做呢個實驗嘅，你又同阿徐仔講嗱，徐仔， 其實我又唔可以話你錯，但係我又唔可以話你啱，點都好啊，俾個解釋我啊。咁其實喺錘仔面前有兩條路，第一條路呢，就係錘仔揀選咗嗰條路，佢就話啦，其實呢，有一啲短暫嘅指令呢，就喺個物種一嘅病部嘅，都暗示咗錘仔呢係堅持自己嘅假說係正確嘅。嗱，遺傳信息真係喺個物種二嘅底部嘅咧，嗱，佢個底部係呢樹噶嘛，應該呢就係啲遺傳信息真係上到去嘅，不過呢，有啲短暫信息喺度阻住晒。而第二個方向呢，就係錘仔就講啦，其實呢，喺個底部呢，就有啲短暫嘅信息，亦都暗示咗話唔定我錯呢，嗰啲遺傳信息呢，可能真係儲存喺個病嗰樹，唔係儲存喺個底嗰樹呢。咁啊，錘仔啦，擇善而固執，佢仍然相信自己係正確嘅。而阿錘仔呢，就保持佢嘅初心，堅定佢嘅信念呢，就覺得自己應該係啱嘅。所以呢，我哋就要睇下實驗4咯。實驗4就點啊？唔理你啦，斬咗呢個頭，再睇下最後結果。最後結果係守得雲開見月明，真係生翻個花花頭出嚟。咁即係話，原來最後尾嘅結果嗰個頂部嘅形態係真係屬於花花形狀，而呢個花花嘅形態係由物種二嘅底部所去決定，而唔係由物種一嘅病部所去決定，亦即係錘仔所講嘅結論係正確嘅。成個 part B 有咩嘅變奏呢？佢可以問下你嘅，有咩辦法都可以測試得到阿錘仔嘅假說係咪正確呢？嗱，而家呢，就係將1號嘅病同2號嘅底結合嘅，下次我反翻轉咯，將1號嘅底同2號嘅病去結合，又睇下會發生咩事啦。照道理呢，都係仍然有一個混雜嘅形態嘅，唔緊要，再做再斬呢個頭，再睇下你個樣。到頭來我哋會發現個頭應該係會變翻圓形頭嘅，因為遺傳物質係儲存喺個藻類嘅底部。經過咁多輪嘅腦震盪呢，終於去到 part C 啦。part C 就問翻啦，阿錘仔做嘅咁多件事，究竟係體現咗啲乜嘢嘅科學本質呢？同埋啦，唔該俾個原因我啦。咁所以啦，成個答題應架呢，你就要答翻。第一，究竟個科學本質係乜嘢？呢度有好多俾你揀噶啦。嗱，成堆科學本質咧俾咗你噶啦。段片咧都有好多同學睇咗噶啦，有好多同學咧都講嘅。原來一段片綜合咗咁多嘅科學探究，綜合咗咁多嘅科學本質咧，聽聽下佢就有個脈絡出現咗。而第2分你就要有個原因俾我啦。咁成個實驗錘仔做咗啲乜嘢呢？嗰啲咩觀察啊、問問題啊、做假設啊，佢做咗幾多個實驗啊？做咗4個，當中佢探究咗啲乜嘢，而亦都有啲咩嘅假說呢？佢想揾嘅係遺傳物質嘅位置同埋遺傳物質嘅表達，究竟係乜嘢嘅一回事咯？我哋就睇下錘仔嘅經歷究竟表現咗啲乜嘢嘅科學本質啦。第一個咧就係講科學知識係建基於或者衍生於一啲對於自然世界嘅觀察啦。咁其實錘仔咧整個實驗都係不斷去觀察翻呢個單細胞藻類嘅頂部有冇再生啊？呢個再生咧係要啲咩嘅條件啊？而根據翻個頂部嘅再生，就會知道翻究竟基因遺傳物質佢嘅位置喺邊度？原來喺個底部。第二啦，科學探究咧係需要創意同埋想像力嘅。你諗下，好人好者咧係真係唔會無啦啦諗到啊，不如斬咗個頭試下，斬咗個頭都未完，不如我試下將佢個病同埋個底部溝埋嚟玩啊！你諗下，錘仔做嘅咁多實驗，都係斬下頭、斬下尾，跟斬咗個頂咧，都仲生得翻，但係斬咗個底就生唔翻咯。啊，代表咩意思呢？跟住到第3啦，就係咧科學咧係一個不斷發問嘅過程。你諗下，錘仔佢成個實驗當初其實就只係想知道基因信息喺邊個位置嘅啫，但喺個過程當中，佢知道咗原來喺呢個基因遺傳物質有一啲短暫嘅指令噶。呢一樣嘢係佢當初冇估到、冇諗到係會發現噶，而家發現咗啦。當然喺佢個年代就未知咩叫信使 RNA 啦，但係當其時佢已經知道遺傳物質係有一啲係永恆嘅，有一啲呢係短暫嘅。而整套嘅概念就係科學本質啦，而經過不斷嘅發問，我哋就建構到現今嘅遺傳學知識啦。好，又嚟到一點出發啦。今次嘅實驗呢，就用遺傳信息作為切入點嘅，考我哋2樣嘢。 邊度儲存同埋如何表達。先講儲存先，儲喺細胞核係我哋往後要學嘅嘢。今次呢個題目係冇講到點解嘅，而呢個概念亦都唔難嘅。我哋喺翻呢個細胞去抽取啲遺傳物質出嚟啦，跟住又睇下啦，跟住我哋又睇下咯，斬咗你個底部啊，斬咗你個頂部啊，究竟有冇再生嘅出現呢？其實都係一個科學探究嚟嘅。而另一款儲存啊，就係有關於 DNA 嘅特性啦。DNA 雙螺旋嘅特性有咩咁特別呢？咁今次我喺度咧輯咗書本嘅概念啦。點解 DNA 係可以作為一個遺傳物質呢？而 DNA 嘅雙螺旋同佢複製 DNA 又有啲咩嘅關係呢？呢個就係適應性特徵啦。遺傳信息嘅表達，轉錄轉譯一定要識噶啦。仲有啲咩嘅概念啊？遺傳密碼啦，三聯體密碼嗰啲，你要學識去睇翻嗰啲咩 AAU 啊、AUG 啊、GGG 啊嗰啲咯。而就將我哋嘅基因遺傳信息嘅表達同製造蛋白質拉上關係啦。而整條題目係用科學探究嚟包裝嘅，就自不然問下你科學探究嗰啲唔同步驟啦。而今次我哋想探究嘅就係一個單細胞藻類嘅再生，同埋佢嘅形態同佢嘅基因信息有咩關係呢？啲基因信息儲存喺邊度呢？啲基因信息點樣表達呢？過程當中我哋都發現咗，原來有啲短暫嘅信息都會影響咗蛋白質嘅製造嘅。2016 question 8 is about science investigation. So in every year you can find those questions. They will show you a scientific investigation. What is the aim? What is the possible result? Any observation? Any hypothesis? Any conclusion? And then ask you about the nature of science. So in this question, we are talking about the scientific investigation held by Hammerling. Hammerling tried to find out where the genetic information was stored inside the eukaryotic cells, and he used some unicellular algae to carry out a series of experiments. So you can see that these two species of unicellular eukaryote, they contain only one cell. You can see the morphology of this one cell is very different from the unicellular organism we learned before. For example, the amoeba. And the shape of this unicellular algae is also very different from the red cell, ribose cell, or mesophyll cells, something like this. And there are foot, stalk, and cap, three parts in this unicellular algae. And there are two species, one with the round-shaped cap, and the other one with the flower-shaped cap. Before we talk about the experiment, let's take a look at the question. You need to describe the result of the above experiment. So in this question, is checking the critical skills, the synoptic skills. How can you directly describe the observation, the results of the treatments 1 and 2? So let's talk about the experiment. There are two treatments 1 and 2. In treatment 1, we remove the cap from the species 1. And the treatment 2, we remove the foot from the species 1. And then we observe if any regeneration occur in the remaining part. And in this question, I would like to talk about another skills is to answer in short sentence. And it is a crucial examination skills. How can you express yourself directly? In this question, we are going to observe, did any regeneration occur in the remaining parts after a certain treatment? So let's see what we have to answer. So let's talk about the scaffolding. We need a comprehensive description. First half, we need the treatment. What did Hammerling do? And for the second half, what is the result? What did Hammerling observe? So we can see that in the treatment X, Y was removed. So you can just substitute the number treatment 1, treatment 2, and which part of the algae was removed. And for the second half, the result is found that or we observed that I would like to talk about that for the so-called model answer, the marking scheme. The sentence structure is too complicated to follow. Those with cap removed could regenerate the cap. Or you can say that the stalk and foot alone could regenerate the cap. Surely, if you read it, you may be able to understand it. But when you are doing the exam paper, how can you formulate such complicated sentence structure? It doesn't make sense. So I need to show you the alternative answer. You answer in short sentence and then express yourself logically and correctly. In the treatment 1, the cap from species 1 was removed, and it was found that the regeneration of the cap occurred. And in treatment 2, the foot of the species 1 was removed, and it was found that the regeneration of the foot did not occur. And based on the results of treatments 1 and 2, Hamelin hypothesized that the hereditary information is stored in the foot of the algal cell. Before we talk about the experiment, let's take a look at the questions. Hamelin concluded that when the cut stalk was transplanted, it contains some short-lived instruction derived from the foot of the species 1, resulting in a mixed morphology of the cap in the first regeneration. And we need to suggest the type of biomolecule that carry the short-lived instruction. And how could the biomolecule suggest in part 1 affect the morphology of the cap? So let's take a look at the experiments. So in treatment 3, Hamelin did the grafting the stalk of species 1 to the foot of the species 2. And after grafting, the cap formed from the first regeneration showed in a mixed morphology. And then he removed the regenerated cap, and the cap formed from the second regeneration looked exactly the same as the species 2. That's why he draws such conclusion. But what is a short-lived instruction? It's checking the concept of the short-lived genetic material, and it is the messenger RNA. Remember to talk about the full name. For the second one, how could the messenger RNA affect the morphology of the cap? This question is checking you the concept about the importance of the messenger RNA. For the messenger RNA, surely we need to recall the process translation. We use it as the template. We use the messenger RNA as the template for the protein synthesis. And we need to relate it to the morphology. So the second concept we are checking is to recall the relationship between the proteins and the morphology. The messenger RNA directs or instructs the protein synthesis through translation. And the protein produced determine the morphology of the cap by acting functional protein such as M sites or the structural proteins. And for part 2, how do the results from this experiment support Hamelin's hypothesis? You need to relate the design of the experiment to the hypothesis proposed by Hamelin. The hereditary information is stored in the foot of the algal cell. And let's take a look at the treatment 3 and predict the possible result. Hamelin did the grafting, the stalk of species 1 to the foot of the species 2. And then if the hypothesis is correct, the cap should look like exactly the same as the species 2 because the genetic information is stored in the species 2. And if the hypothesis is wrong, it means that the hereditary information is stored in the stalk of the species 1. So that means the cap, it should look like the cap of the species 1, right? However, surprisingly, the cap, it shows the mixed morphology. It's so embarrassing. Hey Hamelin, how can you explain that? You have to give me a possible reason to explain the mixed morphology. And in front of Hamelin, there are 2 possible reasons. The first reason is that there were short-lived instructions derived from the foot of the species 1. And it implies that he still keeps the same hypothesis. The hereditary information is stored in the foot of the species 2. The second reason is that there were short-lived instructions derived from the foot of the species 2. It implies that the hereditary information is stored in the stalk of the species 1. However, there are some short-lived instructions from the foot of the species 2, and that it disturbed the final morphology. From the whole experiment, we know that Hamelin kept his own idea. He still believes that he is correct. The hereditary information is stored in the foot of the algal cell. Therefore, we have the treatment number 4. He removed the regenerated cap and then see the second regeneration. And what is the final result? The cap look exactly same as the species 2 in the second regeneration. So we can talk about the result first. The final morphological feature of the cap resemble that of the species 2. It shows that the trait is determined by the food of the species 2, but not the stock from species 1, and which is the permanent heredity information. For the whole part B, what is the possible question variation? It may ask you to suggest another method to test Hamelin hypothesis. What do we do? Just do the opposite idea. We do the drafting, the food of the species 1 to the stock of species 2. And for the possible first regeneration, there should be the mixed morphology cap. And then we remove the cap from the first regeneration and observe the cap in the second regeneration. If the hypothesis is correct, means the genetic information is stored in the food of the algal cell, the cap should be same as the species 1. And if the hypothesis is wrong, the cap should be same as species 2. That's the logic for us to prove our hypothesis. And after the exhausting part A and part B, let's take a look at part C. Give one aspect about the nature of science that can be demonstrated in the above discovery and give a reason to support your answer. So you can check the video about the nature of science. And in this question, what is the scaffolding? 1 mark for the nature of science. And for the second mark, you need to support it with the reason. What did Hamelin do in the whole scientific investigation? And recall the aim of the investigation and any hypothesis being tested. So for this part, it can ask you about making observation, asking action, proposing a hypothesis, a lot. You can see the 4 treatments in the whole scientific investigation. And what's the aim of the investigation? And the whole scientific investigation, Hamelin would like to study where is the genetic material stored and how is the genetic material expressed, so we can do the matching. Scientific knowledge is based on and or derived from the observation of the natural world. Hamelin keep doing observation on the regeneration of the cap and leads to the confirmation of the location of genetic material. Doing science requires creativity and imagination. You can see that Hamelin decided different type of experiment, moving the cap, removing the food, do the grafting, put the stalk and the roots together, test his hypothesis. For the last one, science is a process of ongoing inquiry. Hamelin keep asking questions. You can recall that the original aim of the scientific investigation is to find out the location of the genetic material. And throughout the ongoing inquiry, it needs Hamelin to know something about the short life instruction. At that moment, we don't know what is the messenger RNA. At that moment, Hamelin did a very insightful conclusion. There is short life instruction of the genetic material. Let's talk about the curriculum mapping. This question, it starts from the genetic information and it tests us 2 concepts: the storage of the genetic information and the expression of the genetic information. About the storage, of course, we know that the genetic material is stored in the nucleus. But for the storage of the genetic information, we talk about the DNA. Why DNA is well suited to be the genetic material storage? What about the DNA double helix and how is it so important for the DNA replication? And about the expression of the genetic information, we talk about transcription and translation. And we need to recall the concept about the genetic code, the trinap code, that means the codon, and relate it to the protein synthesis. For the whole question is about scientific investigation. You need to recall the basic steps in the scientific investigation. And in the whole question, we are studying the location of the genetic material, how does the genetic material affect the protein synthesis, and how do the genetic materials, how does the short life instruction, the messenger RNA, affect the protein synthesis, and in turn to affect the morphology of the algae cell. And it shows different aspects of the nature of science.